Okay, in this video we are going to discuss your post-fertilization event. Before this, a quick recap. What we have understood, this is your vegasporangium, right? And this is your, say for example, ये क्या है भाई आपके nucellar cell. Okay, nucellar cell. Out of this, one become functional, right? ये functional है. That is your one is differentiated into megaspore mother cell. Now this megaspore mother cell, it is going to undergo what? Meiosis. So at the end of meiosis, you will get 1, 2, 3 and 4 haploid megaspore. Out of this 4, 3 degenerates and 1, can you see this? Yes, this 1 becomes your functional megaspore. So ये जो green वाला है वो क्या है आपका? 1 functional megaspore and ये उसका nucleus. Okay, now what I have taught you, this nucleus will undergo 3 mitotic division in succession, in the sense एक के कितने बनेंगे? 2, right? These 2 are nucleus, not the cell. This nucleus goes towards the opposite end, right? Then second mitotic division forming 4 cell. Then third, your mitotic division resulting in formation of 8 nuclei. 1, 2, 3, 4, 5, 6, 7 and 8. So ये जो 8 है वो क्या है आपके न्यूक्लियाई. यहाँ पे साइटोकाइनेसिस इज स्टिल नॉट डन. ऑफ दिस 8, 2 polar nuclei comes in the center, जिसको हम कहते हैं polar nuclei. This is called as your polar nuclei, followed by which there occurs cell wall deposition. So this is your one cell, this is your second cell, this is your third cell, जिसको आप कहते हैं antipodal cells. This combines to form your central nucleus. This one is your egg cell. This two represents your synergid. तो इसको आपने बोला egg apparatus, followed by which we have understood pollination, right? Pollination हो गया. Say for example, ये क्या है बेटा जी? आपकी pollen tube, and this pollen tube carries your 1 and 2 male gamete. Along with this, there is presence of 1 tube nucleus. This tube nucleus degenerates. This pollen tube is going to enter one of your synergid through filiform apparatus, followed by which pollen tube will burst, releasing 2 male gametes within this embryo sac or within the cytoplasm of synergid. This we have already discussed. Okay, so now inside synergid there is presence of your 2 male gametes, of which one male gamete fuses with, we very well know that one male gamete fuses with egg to form your diploid zygote. ये क्या बनाएगा? Your diploid zygote. And remaining one, can you say this? Yes, this remaining one male gamete goes with your central nuclei, to form your primary endosperm nucleus. In your textbook it is written primary endosperm cell, one and the same. Okay, so एक मेल गैमेट फर्टिलाइज करे एग को, दूसरा मेल गैमेट फर्टिलाइज करे सेंट्रल सेल को, रिजल्टिंग इन फॉर्मेशन ऑफ जाइगोट, जो कि होता है डिप्लॉइड. एंड प्राइमरी एंडोस्पर्म न्यूक्लियस बोलो या प्राइमरी एंडोस्पर्म सेल बोलो, जो कि होता है ट्रिप्लॉइड. मोस्ट प्रोबेबली ये जो एंडोस्पर्म होता है इज ट्रिप्लॉइड इन नेचर. In certain cases it may be haploid, tetraploid or many more. Okay, लेकिन most probably आपका PN जो होता है it is triploid, उसकी प्लॉइडी होती है 3N, that is your triploid. Okay, now we are discussing, ये हो गया आपका डबल फर्टिलाइजेशन. नाउ वी आर डिस्कसिंग पोस्ट फर्टिलाइजेशन इवेंट. फर्टिलाइजेशन हुआ, इसके बाद क्या? ओके, सो वी आर हैविंग 2 थिंग्स रेडी एज आई टोल्ड यू, दैट इज योर zygote and your primary endosperm cell. Now this zygote develops into your embryo. It develops into your embryo, and this primary endosperm cell undergoes cell division of course to form your endosperm. अब ये endosperm क्या है, मैंने आपको बोला था, this endosperm is your nutritive tissue. It is nutritive tissue which provides your nutrition to डेवलपिंग एम्ब्रियो. ओके, ये किसको न्यूट्रिशन प्रोवाइड करता है? टू डेवलपिंग एम्ब्रियो. तब आप मुझे बताओ किसका डेवलपमेंट फर्स्ट होना चाहिए? एंडोस्पर्म डिवीजन फर्स्ट होना चाहिए या जाइगोट डिवाइड फर्स्ट होना चाहिए? ऑफ कोर्स देयर अकर्स फर्स्ट division of your primary endosperm nucleus. What is the reason for the same? क्योंकि ये न्यूट्रिशन नरिशमेंट प्रोवाइड करता है फॉर टू डेवलपिंग एम्ब्रियो. इसकी वजह से फर्स्ट देयर अकर्स डिविजन ऑफ योर प्राइमरी एंडोस्पर्म सेल. After some time there will be division of your zygote. So this events we are going to understand in post fertilization event, that is your endosperm development number 1, and second we are going to understand embryo development. Okay, now let us begin with first event of post fertilization, that is endosperm development. We very well know that endosperm development precedes embryo development. मतलब embryo development के पहले क्या होता है? There will be division of your primary endosperm nucleus which results in formation of your endosperm tissue. Okay, so now this PEC, that is your primary endosperm cell, which is formed by fusion of male gamete and your secondary nucleus or your central cell, it undergo division. Of course ये division कौन सा होगा? Mitotic division, which results in formation of endosperm tissue. What will be the ploidy of this endosperm tissue? इसकी ploidy क्या है? 3n, that is triploid. तो but of course मैंने आपको बोला कौन सा cell division होगा यहाँ पे? Mitosis. So there will be formation of your triploid endosperm tissue. वापस से एक बार, endosperm का काम क्या है? It provides nourishment, nutrition to developing embryo. Okay, now this endosperm development is divisible into 3 parts. मतलब वो 3 तरीके से हो सकता है. We are naming it accordingly. Nuclear type, second one is your cellular, and third one is your halobial. Okay, first type is your, say for example यहाँ पे आप A लिखो, nuclear type, this is your cellular type, and this is your halobial type. So these are the 3 types of your endosperm development. Okay, now we are beginning with your nuclear type. As the name suggests, यहाँ पे कुछ nucleus का हो रहा होगा. Okay, now look, in nuclear type of endosperm development, what happens? First division and several other divisions are unaccompanied by wall formation. इसका मतलब क्या होता है? Karyokinesis occurs but there will be no cytokinesis, जिसकी वजह से cell wall deposition नहीं होता. In the sense only and only nucleus divides, there will be no deposition of cytoplasm. Okay, इस वजह से इसको बोला जाता है nuclear type. Now the nuclei which are formed, okay, the nuclei which are formed either remains free, या तो वो फ्री रहे, or in later stage they may get separated by wall. Later stage in the sense, if a fruit attains maturity, right, after maturity there is possibility कि cytokinesis हो जाए. तो ये क्या है बेटा जी आपका न्यूक्लियर टाइप. व्हाट इज न्यूक्लियर टाइप ऑफ एंडोस्पर्म डेवलपमेंट? इन विच ओनली न्यूक्लियस डिवाइड्स, देयर इज नो डिपोजिशन ऑफ सेल वॉल. मतलब कैरियोकाइनेसिस के बाद साइटोकाइनेसिस नहीं होता, जिसकी वजह से यू विल गेट many free nucleus. Sorry. Okay, this nucleus may remains free or in later stage they are separated by wall. Okay, now say for example here I am drawing your embryo sac. What is this? This is your embryo sac. इसको आप female gametophyte भी बोल सकते हो. Okay, now this is your embryo. ये क्या है बेटा जी आपका embryo, and where embryos are located? Of course at your microphylar region. So this is your microphylar region and this represents your चलाजा. ये क्या रिप्रेजेंट करता है आपको? चलाजा. तो ये क्या बनाया मैंने? एम्ब्रियो सेक. नाउ दिस इज द this is your PEN, that is your primary endosperm nucleus. Now what happens in nuclear type of endosperm development? This undergoes karyokinesis. मैंने आपको बोला cell wall deposition नहीं होती, right? तो if I want to show once again, this is your embryo sac. Okay, this is your embryo, this is your micropylar end, and this is your chalazal end. Okay, now this nucleus will undergo mitotic division to form many free nuclei. This results in formation of many free nuclei. Okay, as दिस डिवीजन प्रोग्रेसेस, जैसे जैसे ये डिवीजन हो रहा है वैसे वैसे व्हाट हैपन्स, दिस फ्री न्यूक्लियस आर पुश्ड टुवर्ड्स पेरिफेरी. दिस फ्री न्यूक्लियाई आर पुश्ड टुवर्ड्स पेरिफेरी, creating central large vacuole. Am I clear? जैसे जैसे ये न्यूक्लियस डिवाइड हो रहा है वैसे वैसे they are pushed towards periphery, forming a thin layer and creating central large vacuole, as you can see over here. Okay, तो ये peripheral region में thin layer is of your free nuclei. Most probably they are aggregated towards your micropylar region and chalazal region. ठीक है, most of them they are aggregated towards microphylar region or your chalazal region, and generally the nucleus which are present at your chalazal region पे जो आपका न्यूक्लियाई होता है, ओके, दिस न्यूक्लियाई आर लार्जर देन द न्यूक्लियाई प्रेजेंट एट योर माइक्रोफाइलर रीजन. सिंपल एज दैट. यहाँ पे जो न्यूक्लियस होते हैं वो कैसे होते हैं? लार्जर इन कंपैरिजन टू दैट present on your micropylar region. Now what happens, say for example if we are talking about coconut, तो नारियल का पानी जो आप पीते हो that is nothing but your nuclear endosperm. The tender water of coconut is nothing but your nuclear endosperm. वो न्यूक्लियाई पीते हो आप. Okay, now what happens as fruit matures, if you are talking about coconut, okay, as coconut matures what happens, ये जो peripheral nuclei है, ये जो से for example I am representing few over here, इसके surrounding क्या होगा बेटा जी? cell wall deposition. Can you see this? इसके आजू बाजू क्या होगा? There will be deposition of cell wall. Am I clear? And this what is your, वो जो कोकोनट आप खाते हो white वाला portion, okay, that is your coconut cellular endosperm. What is that? That is your cellular endosperm. Am I clear? Once again मैंने क्या बोला आपको? ये embryo sac है. This is microphylar region, this is chalazal region, this is embryo, and this is your primary endosperm cell या primary endosperm nucleus. In the nuclear type what happens? This undergo कैरियोकाइनेसिस, राइट, सेल वॉल डिपोजिशन नहीं होती, which results in formation of many free nucleus. This free nucleus moves towards periphery, as you can see over here, creating central large vacuole. Okay, most probably this nuclei aggregates at your micropylar region and your chalazal region. मैंने important point आपको ये बोला कि free nucleus of your chalazal region are larger than that of your micropylar region. Okay, as your fruit matures, अगर आप कोकोनट की बात कर रहे हो, as your coconut matures, this nucleus gets cell wall deposition, that is your cellular endosperm. वो क्या है आपका cellular endosperm, second type that I have written. Okay, so this is your nuclear type of endosperm. Okay, now let's talk about your second type of endosperm development, that is your cellular type. This is simple thing. In this thing, karyokinesis followed by cytokinesis, simple as that. मतलब nucleus का division होता है, followed by there will be formation of wall. Okay, so in this first and most of following divisions are accompanied by wall formation. Okay, are accompanied by wall formation, and this results in formation of several chambers within the embryo sac. इसकी वजह से your embryo sac appears several chambered. Okay, now ये जो आपका division है, the first wall division is usually कैसा होता है, transverse, is usually transverse, whereas in certain case it may be vertical or it may be oblique also. Once again मैंने आपको क्या बोला, cellular type में क्या होता है, simple as that, your karyokinesis followed by cytokinesis. Most probably सारे division ऐसे ही होते हैं, लेकिन in certain case karyokinesis occur which is not immediately followed by cytokinesis, जिसकी वजह से there will be presence of more than one nucleus in particular cell. Okay, so in this your first, मैंने आपको most of बोला है, सभी नहीं बोला, most of your cell division is accompanied by wall formation, and thus sac is divided into several chambers, highlight this point, some of which contains more than one nucleus. Okay, ये wall formation या तो transverse होती है, या तो vertical होती है, या तो वो oblique होती है. Say for example, if I want to draw over here, once again this is your embryo sac, micropylar region, chalazal region, यहाँ पे आपका होगा embryo, and this is your primary endosperm cell. Okay, how cell division occur, देखो, this is, I am dividing this cell transversely. Okay, I am dividing this cells transversely, resulting in formation of two cell. Am I clear? This is case number one. In certain case, once again ये आपका primary endosperm cell है, right? It divides vertically like this, which results in formation of your, once again, two cells like this. Whereas in certain case, वापस से यहाँ पे आ जाओ, ये क्या ड्रॉ करा मैंने आपका primary endosperm nucleus, it divides obliquely, मतलब like this, which results in formation of your first cell to look like this. तो ये transverse, ये vertical and ये oblique. Transverse, this is your vertical and this is your oblique cell wall formation. Simple as that. तो ये क्या हो गया आपका cellular endosperm. Once again let me remind you, in coconut what you are drinking, tender coconut water, is free nuclear endosperm, and white kernel which we are eating is your cellular type of endosperm. Okay, the last type of endosperm development is halobial type. Actually, you can say it as a subtype of your cellular endosperm development. Okay, this halobial type of endosperm development is intermediate between nuclear and your cellular type. Most important thing, halobial type, right, वो intermediate है between your nuclear and cellular type of endosperm development. Now in this type what happens, जो first division होता है, okay, the first division is like your cellular type, मतलब karyokinesis followed by cytokinesis which resulting in formation of two cells. First division is like your cellular type, मतलब the first division is followed by transverse wall formation. ट्रांसवर्स वॉल फॉर्मेशन जिसकी वजह से आपको दो चैम्बर्स मिलेंगे, वन चलाजल एंड सेकेंड माइक्रोपाइलर चेम्बर. ओके, देखो यहाँ पे वंस अगेन दिस इज योर एम्ब्रियो सेक, जाइगोट, प्राइमरी एंडोस्पर्म सेल. नाउ व्हाट हैपेंस, फर्स्ट डिवीजन जो मैंने आपको बोला वो कैसा होता है, लाइक योर सेल्यूलर टाइप, मतलब कैरियोकाइनेसिस फॉलोड बाय cytokinesis which results in formation of two cell. This is your cell number 1 which is located at your chalazal side, and say for example this is your cell number 2. okay, ये 1 and ये 2 which is located at your micropylar side. okay, so this is your chalazal chamber and this is your micropylar chamber, followed by which जो division होगा it will be like your nuclear type, मतलब followed by which what will happen, this nucleus will undergo division to form many nuclei, and most probably this nuclei are formed at your micropylar chamber. Once again, ये प्राइमरी एंडोस्पर्म सेल है, इट विल अंडरगो डिवीजन टू फॉर्म योर two chambers, two cells at chalazal and micropylar region, followed by which what you will get, this is the free nuclei at your chalazal end and this is the free nuclei at your micropylar end, and most probably this free nuclei are present towards your micropylar chamber. So this is the type of your endosperm development. Number 1 is your nuclear, second one is your cellular, and third one is your halobial type.